Hello students, welcome to Studywell e-learning, and today we are going to discuss about a question in transcription, and that question is why both strands of DNA are not transcribed, or why both strands of DNA are not copied as RNA. Okay, so this is our question for today. This is our topic for today, and this video is a kind of a remake. I have already made a video on this topic, but that video is in Hindi, and that's why I'm going to make this video in English. Okay, so I hope it's going to benefit you. So let's get started. Question is why both strands of DNA are not transcribed. Why DNA both strands are not copied. So this question has two answers. There are two reasons for this process to happen. So first is, let's discuss both of the reasons. First reason is that if both strands of DNA gets copied, then the strands will be coding for two different proteins. So let's get into details. First, let's see what does a DNA do. A DNA codes for RNA. RNA gets transcribed from DNA, and then this RNA codes for proteins. This RNA gets a code that helps to make proteins. Okay, this is called central dogma. You know, this process is known as transcription, in which RNA is formed from DNA, and the second process is translation. This is translation and transcription, and together we call it central dogma. Okay, so it means DNA is coding for proteins through RNA. Okay, and then these proteins may act as enzymes. And enzyme is responsible for the functioning of the cell. Means they give this metabolism. They are responsible for metabolic reactions. So if enzymes are formed, only then metabolism can be carried on. And this metabolism, here the reactions, some products will be formed due to the presence of these enzymes. And what is the result? There will be a phenotype. So, how the genes give phenotype? This is a gene, and this gene gives a certain phenotype by passing through these stages. Okay, gene helps to make RNA, then protein, and then these proteins may act as enzymes, and enzymes give phenotypes. That's why we say DNA gives phenotype. DNA is the master copy for formation of proteins in the cells. Now, what is our question? Question is why both strands of DNA are not transcribed. Now see, this central dogma here, let's make an imaginary DNA strand, then we'll come to the answer to this question. So there is a, suppose this is our DNA. As you know, it has got two strands, and both strands are anti-parallel to each other. Two strands are anti-parallel. One strand runs from 5' to 3' direction. The other strand runs from, it's in opposite direction, 3 to 5. Okay. Yes, strands, these are anti-parallel to each other. And these strands, suppose here on this strand of DNA, this is a particular sequence. You know there are 4 nitrogenous bases in a DNA. So G, 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 C, C, A, T, G, C, C, C. Suppose this is the sequence on this particular strand of DNA. So what will be the sequence on the other strand? You all know that both strands, two strands of DNA are complementary to each other. Always remember, they are not identical, but they are complementary to each other. So on the second strand, it will be C, 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 G, G, T, A, C, and then G, G, and G. This will be the sequence, and the two strands are held together by hydrogen bonds. This is a DNA. Now this DNA, suppose both strands are making RNAs, So what will happen? Here the DNA will form, this strand of DNA will form, here see, this is only imaginary thing. Later on you will come to know about two strands in this DNA. One is coding and the other is template. And then you will know to which strand the newly formed RNA is complementary and to which strand it is identical. That you will know later on. But before that, we are just making two RNA strands out of it, and both strands I am going to make here will be complementary to the strand from which they are being drawn here. So here this strand will code for an RNA. The codon on this is coded languages GGG. Sequence of nitrogenous bases is GGG, then CC. So what will it be on RNA? It will be CCC, then GG. For A, it is going to be U, as we are making complementary strands. Instead of A, we will write U. So the strand, which is this DNA strand, which is coding for RNA, here instead of A, it will be uracil. Then T, it will be adenine. Then C, G, G and G. Okay, this is one strand of RNA formed from one strand of DNA. Suppose the other strand is also being copied. Okay, that will come to it later on. But let's see what happens to this strand. Now, this RNA is formed inside the nucleus. Now this RNA, this is RNA. This RNA, mRNA, will go to the cytoplasm and there it will help in protein synthesis. How it is going to help in protein synthesis? It is acting as a slip of information on which the information is written in a coded way, in a coded language. So this language will be cracked with the help of tRNA. Now, what is the coded language? You know, there is a codon. One codon codes for one amino acid. Proteins are made up of amino acids. Now, what will happen? 3 nitrogenous bases. They will make a single codon, and one codon is going to code for one amino acid. Suppose this is CCC. You know, there are 64 codons. Okay, out of the 64, 61 code for amino acids. So here we have a chart, full chart. Here, this is a chart. This chart shows all the codons. Okay, with the help of this chart, we can check which amino acid is going to be present at which position in a polypeptide chain. So here, this CCC codon is going to code for a certain amino acid, say amino acid number 1. And this GGU is going to code for another amino acid. Let's call it AA2. And ACG is going to code for a third amino acid. Let's call it AA3. And these will be linked together by peptide bonds. Okay, peptide bonds will be formed between amino acids. And now there are many such amino acids in this chain. So this is going to code for 1, 2, 3 and 4. Let's give it another nitrogenous base. So there will be 4 amino acids linked together. So this is known as a polypeptide chain. So we will get a polypeptide chain. This polypeptide chain is formed by this mRNA with the help of amino acids, with the help of ribosome, and the information mRNA got from DNA. Okay, so this is a certain polypeptide chain. Let's call this polypeptide chain as A. Let's name it A. A polypeptide chain. This is the name of the chain, A polypeptide chain. And now let's go to DNA again. If this strand of DNA is also transcribed Okay, we are taking a problem. We are just talking about if this happens, then what is the result. Okay, actually it doesn't take place in nature, doesn't take place inside the cell. So let's now make RNA out of this DNA strand. As you know, DNA strands are not identical, they are complementary to each other. Now, if this strand gets transcribed, we will get, अब हम इसका complementary बनाते हैं. Let's make an RNA that is complementary to this strand of DNA. Let's make it. So it will be GGG, then it will be CC, A, and then U, G, C, C, C. Okay, so it will be from, let's give it a direction. If this is given 3' to 5', because we have made complementary to this, so we will give 5' to 3' direction to this particular RNA. So this is also mRNA. It is also going to code for amino acids. It is going to also going to act as what? As a bundle of coded information that helps to make proteins. It is going to code for amino acids. Again, it has got codons. So these 1, 2, 3, 4 codons will be coding for 4 amino acids. But these amino acids will be different from these ones. Okay. If this chain is having any codon similar to the codon in this mRNA chain, then that amino acid will be the same, but sequence will be different. So it is going to make a different polypeptide. Okay, so let's say this is coding for amino acid number 5, then amino acid 6 is linked to it, amino acid 7 is linked to it, okay, amino acid 8 is linked to it. And this is another polypeptide chain. Let's call it polypeptide chain B. Okay, B polypeptide chain. Done? Now, what is the result? What is the conclusion? The conclusion is that single DNA strand is coding for 2 different polypeptides. Okay, if both strands of DNA are transcribed, then the DNA will end up coding for 2 different polypeptide chains, and this will complicate the gene expression. Okay, because gene expression means, see, I just told you that proteins act as enzymes and they are responsible for giving phenotypes. So, which protein will be giving phenotype here? This gene, this particular gene is giving B phenotype or A phenotype. That will be a confusion for the cell. Okay, it will be very difficult for the cell to deal with this. So, this is a problem for the cell. This will complicate the gene expression, because which phenotype is to be given by this particular gene? One gene can give only one phenotype. One gene can give only one, it can code only for one polypeptide, not for 2 different polypeptides. That's why transcription of one DNA chain is prevented by the cell. Only one chain is being transcribed. Okay. So this is the first reason. What is the answer to the first answer to this question? This is that if both strands of DNA are transcribed, then the 2 strands formed by the 2 RNA strands formed will be coding for 2 different proteins. Okay, this is the answer to this question. This is the first reason why both strands of DNA are not copied as RNA, why both strands of DNA are not transcribed. This is the one reason. But the 2 RNA strands formed from one DNA will code for 2 different polypeptides. Okay, and this will complicate gene expression. So this complicates gene expression, and gene expression means the appearance of phenotype. Okay, this is one reason. For this reason you should know the central dogma. Okay, and second reason is simple as compared to this one. Second reason, now you know the two strands of DNA are complementary to each other. Always remember, they are not identical but they are complementary. Let's take GGCC, ATC, CCGG, TAG. These two strands are not identical but they are complementary to each other. Okay, so these strands, if both strands are being transcribed, will they be identical? No. Will they be complementary? Yes, they will be complementary. The two RNA strands formed out of it. Let's make two RNA strands. One strand will be GGCCAUC and the other strand formed will be CCGGUAG. Now check, aren't they complementary to each other? If DNA strands are complementary to each other, the RNA strands transcribed out of them will also be complementary to each other. So what will happen? There will be hydrogen bonding between the complementary nitrogenous bases. Guanine will bond with cytosine, guanine. Okay, and adenine will bond with uracil. So what will happen? What will we get? We will get a double-stranded RNA. The result is formation of a double-stranded RNA. We will get a double-stranded RNA as a result of transcription of both strands of DNA. So this dsRNA is kind of locked. It cannot be translated. Their translation cannot occur here. So no translation will take place due to the presence of a double-stranded RNA. Because double-stranded RNA is formed, will not be transcribed. Not be translated. So no proteins will be formed. No proteins means no enzymes. No enzymes means no phenotypic expression. No phenotypic expression, then what is the use of this DNA? What is the use of this transcription process? Okay, why this transcription occurred when the RNA produced was not going to be translated? Then what is the use of this process? The transcription is of no use. Okay, so now you understood that if both strands of DNA are transcribed, then they will code for two strands of RNA which are complementary to each other, and these two RNA strands will form a double-stranded RNA, which that double-stranded RNA is not translated. So the process of transcription becomes a wasteful process. Okay, so this is the second reason why both strands of DNA are not transcribed or are not copied as RNA. Okay, so this is the second reason. Let's write the second reason also. The RNAs formed or transcribed from two DNA strands will form a double-stranded RNA as they are complementary to each other. Okay, and they are not translated. So this prevents translation. Translation and transcription will be wasted. It will be a wasteful process. So these are the two reasons why both DNA strands are not transcribed. Okay. So I hope you have understood. So thanks for watching and do subscribe to my channel. Thank you.